Okay, so here's where we were last time. Um, I was talking about the connections. We've gone through the photoreceptor, bipolar, and ganglion cells. Talked about a little bit the lateral geniculate and the thalamus. That's going to send connections onto visual cortex. Visual cortex is going to start perception. From here, we're going to outsource to other parts of the brain that I'm not going to go into as much detail about, but you need to know exist. All right, so right now, I want to distinguish between your visual field versus receptive field. Okay, so this is something students often get confused about. You need to be able to keep these straight in your mind or you will get confused. The visual field is everything you can see. Okay, so everything you can see is the visual field. Receptive field is going to be that part of the visual field to which an individual cell responds. Okay. Now, when we're talking about receptive fields and you're analyzing receptive fields, one of the things you have to be clear about is which cell you're talking about. Okay, so in general, the photoreceptor has the smallest receptive field. Several photoreceptors are going to hook up to a bipolar cell. So if light hits any of, say, the 3 or 4 photoreceptors that the bipolar cell is hooked up to, the bipolar cell will respond. Ganglion cells have even larger receptive fields. And then when we get into cortical cells, these have even larger receptive fields. Okay, so going from smallest to largest receptive field. So this diagram sort of shows you that. If you look at an individual photoreceptor, so everything here is the visual field. If we take a point of light, just a very tiny point, that is going to hit one photoreceptor. Okay, so if it hits that photoreceptor, right, that place in space is that photoreceptor's receptive field. Now, if I move this light, let's say I look at a point of light here, this receptor will no longer respond because the light won't strike it. It'll strike a different photoreceptor, and it'll be in a different photoreceptor's receptive field. Okay, but if we're focusing on the cell here, only a point of light at this point in space will have an effect on the photoreceptor. Now, if we have a few photoreceptors linked to, say, one retinal ganglion cell, like in this diagram, now if light strikes any of those photoreceptors, the ganglion cell will respond. Okay, so if light is here, or here, or here, or here, my ganglion cell will respond. Okay, so by having multiple cells connect to an individual cell, so multiple photoreceptors connecting to a retinal ganglion cell, we're going to get a larger and larger receptive field. Okay, so visual field is everything you can see. The receptive field is the point in space to which an individual cell responds. The receptive field is different depending on which cell we're talking about. All right, so here is another way to illustrate that. So light has to strike the photoreceptor in order for us to respond. In this setup, light strikes this photoreceptor. Okay, if light hits this, right, that's going to be the receptive field in of this photoreceptor. Now, the way it's drawn out here is we've got one photoreceptor to one bipolar cell to one retinal ganglion cell. This is the way it is near the fovea, where we have the highest resolution. Okay. If light strikes this photoreceptor and this ganglion cell is firing, we know very precisely in space where that point of light is. Okay, we've got a very small pixel size, high resolution. That's what we see at the fovea. In the periphery of the retina, it's a little bit different. Here you can see we've got 6 photoreceptors linked up to a few bipolar cells, linked up to one ganglion cell. Okay, so if we look at the receptive field of this photoreceptor, it's not any bigger. It's the same size as it is for an individual. But because we have 6 of them, right? If light strikes anywhere from here to here, anywhere in here, this ganglion cell will fire, will have a response. Okay. Now, the advantage of this is that we can detect low levels of light anywhere in this receptive field. That's good. The downside is we don't have great resolution. So if this ganglion cell right here fires, where's the light? It could be here, it could be here, it could be here, it could be anywhere in here. We don't have a real precise location for it. That's different than up here, right? If this ganglion cell fires, we know within a small space where that point of light is. Okay, so again, the photoreceptors have the same size receptive field. The ganglion cells, though, this ganglion cell has a small receptive field. This ganglion cell has a larger. This is good for low level of light. This is good for high resolution. Okay, so there's a trade-off. All right. Now, if we talk about retinal bipolar cells and retinal ganglion cells, there's an arrangement called on-center, off-surround receptive field. So you know what a receptive field is now, right? It's an area in space. So if we are talking about a bipolar cell, which we are right now, we're going to focus on this cell. And you can see that this cell is going to be hooked up to a whole bunch of photoreceptors. Now, the arrangement is such that the photoreceptors it's connected to in the center of its receptive field are going to be excitatory. Those in the surrounding area will be inhibitory. Okay, so if light strikes these photoreceptors here, that is going to excite this bipolar cell. And that bipolar cell then will release neurotransmitter, excite a retinal ganglion cell, and send action potentials. If, however, it strikes these in the periphery, the yellow ones, right? If we strike these, now we're going to inhibit the bipolar cell, which means a hyperpolarization, less neurotransmitters being released, and fewer action potentials from the retinal ganglion cell. Okay. So again, these hexagons here all represent the receptive field of that bipolar cell. If light strikes here in the purple in the center, we're going to get excitation. That's why it's on center. In the surround, if we hit that, we're going to get inhibition. That's why it's called off. Okay. Now, this is the diagram from your book. And essentially, I want to show you there's two different setups we can have. Okay, so here we're looking at the bipolar cell in this column. In this column, we're looking at the response of the retinal ganglion cell. Okay, here we're looking at an on-center, off-surround setup. Okay, so remember, this is the receptive field of the bipolar cell and the ganglion cell. In this case, with no illumination, nothing at all, we're going to be at rest in the bipolar cell. It's just hanging out. Nothing's going on. In the ganglion cell, each of these lines represents an action potential. So here we are, no light, just hanging out in the dark for whatever reason. And here you can see those retinal ganglion cells are firing action potentials. Okay, so in the dark, baseline, we're at rest, but my ganglion cell is still firing some action potentials. Okay, so this is an important property of all neurons. Neurons are inherently active. And by that, what I mean is that neurons like to fire action potentials. They have a spontaneous firing rate. So even when there's no stimulation at all, nothing going on, neurons are going to fire some action potentials. Okay, and so that's what's going on here in the retinal ganglion cell. Our bipolar cell, again, at rest, but our ganglion cell, every once in a while, just because, fires off an action potential. Okay, your body is going to get information. Sorry about that. Your body is going to get information from changes. Okay, so info is from changes. This is key, from changes in the firing rate. By firing rate, I mean action potentials, right? So an increase in firing means something. It means something's going on. But a decrease in firing is also meaningful. That also means something. Okay, so what you are looking for, what your whole nervous system is geared toward detecting, are changes. Okay, so that's a key concept that we haven't really talked about yet. All right, so here we are in the dark. Now, if we look at the receptive field and measure the membrane potential or number of action potentials going on during a light stimulus. So this represents the light. So we shine a light. We're going to shine it in different places. If we have an on-center, off-surround cell, and we shine a light only on the center, we're going to get a depolarization in the bipolar cell. When the light goes off, we return to rest, of course. Now, this excitation in the bipolar cell is going to lead to a lot of action potentials from the ganglion cell. Okay, so here we are just at baseline firing some action potentials. The light comes on. It's in the center of the receptive field. We get a massive increase in the firing of action potentials. The light gets turned off. We return to normal. Okay, so remember, photoreceptors and bipolar cells don't have action potentials. Only the ganglion cells do. That's where the action potentials start. So depolarization, lots of action potentials. Now, if we only illuminate the surround, okay, what's going to happen is we get a hyperpolarization in our bipolar cell. That hyperpolarization, right, it's going to lead to less neurotransmitter being released, of course. And instead of being at sort of our spontaneous firing rate, when the surround is illuminated, we're going to have a massive decrease in action potentials. So we go from some to none. Okay. In the case where we illuminate the entire receptive field, okay, so both are, we're going to have some excitation, we'll have some inhibition. What ends up happening is we get a slight depolarization in the bipolar cell, but this really doesn't do much. If we look at the number of potentials, we'll get a slight increase, okay? But it's not clearly the same as when it's only in the center, and very different when it's only in the off, okay? So in this case, you can think of it excitation minus inhibition. Excitation wins by a little tiny bit. Sorry about the dog there. All right, so now here we have a different retinal ganglion cell with a different arrangement. Here we have off center, on surround. So now, in this case, as the name implies, when light hits the center, it's going to inhibit things. When light hits the surround, it's going to excite things. Okay, so again, we're looking same setup, bipolar cells, retinal ganglion cells. You're familiar with this. If we shine light only on the center, we're going to get a hyperpolarization, and we are going to get no more action potentials. If we shine only on the surround, we're going to get excitation. We'll get an increase in action potentials. If we shine the whole thing, in this case, the center wins. We get a little bit of a hyperpolarization, so we get a slight reduction in action potentials. Okay, so that's how these work. Now, someone in consultation actually asked me, how does this help? Like, so what? So one thing it's going to let us do is locate things in space precisely. If the light is here, right? If we get an increase in action potentials, I know exactly where that light is. Okay, the other thing you have to remember is we have hundreds of thousands of retinal ganglion cells. So we're going to put these things together to make shapes. Okay, so vision is geared toward two things: detecting edges. That's how you see an object, right? Is by detecting its edges, and detecting movement. Okay, these are two key things. This is going to allow us to see objects. This is going to allow us to escape predators and hunt. Okay, so the visual system really is geared toward these two things. So keep that in mind. All right, so that being said, let's say I have, we'll do a center on, surround off. Okay, just one. If this is excited, right, I get an increase in action potentials in the ganglion cell. My brain knows that when that retinal ganglion cell is increasing the firing rate, here's where the point of light is. Okay, that's some useful information. But the thing to remember too is we're not talking about one. We're talking about many with overlapping views. Okay, so if you look, now we can start to put some things together. If you look at this and we sort of draw this, right? This is going to help us detect a bar of light. Okay, and so a bar of light is basically a line. This is going to help us detect edges. So when we get to the cortical cells, talking about simple cells, complex, hypercomplex cells, you have to remember that those cells are getting input from many retinal ganglion cells. So we can essentially, I'm just going to draw the centers. If we had the centers like this, right? Now you can see how that can form a bar. Okay, and so we're going to use that ability to detect and respond to a bar of light to detect edges of objects and things. Right, so that's a little bit bigger picture type thing. Okay, so again, we've got center on, or sorry, on center. We also have off center. Okay, there's lots of different variations. And the way this works is here we've got our photoreceptors. Okay, these are going to detect the light. Here we've got our bipolar cells. Okay, and so some of the connections are going to be excitatory. So if you look, this is excitatory. The connection here is going to be inhibitory. Okay, so again, it gets very complex, where some connections are excitatory, some are inhibitory. All right. This is, again, just sort of showing you another way to look at it. If this slide helps you, you can look at it more on your own. All right. Now, in the retina, our book talks about several different types of ganglion cells. Again, you have to remember where you are. We're at ganglion cells. Okay. Talk about M, P, and K cells. Okay, these are going to be short for magnocellular. These will be parvocellular. And this will be coniocellular. Okay, so magnocellular, these are large cells. Okay, parvocellular are going to be smaller cells. Conios are going to be sort of a mixture, right? But these cells are going to do different things. If you look, most of the ganglion cells you have in your retina are going to be these parvocellular cells. These are about 70%. These provide detailed information about objects, including color. Okay, so this, you would think maybe a lot around, say, the fovea, right? With cones, hooked up to cones and things like that. These respond to color, red, green mostly. These go in the lateral geniculate nucleus of the thalamus. There's different layers. Okay, your brain keeps information separate until we get to the cortex, where we start to integrate it. All right, so these parvocellular ganglion cells are going to go to parvocellular layers in the lateral geniculate nucleus. These are small cells. These are slow in terms of transduction and processing. These have a small receptive field size. Small receptive field means low sensitivity, but high resolution. That's good. That's going to help us see objects. Sensitive to high contrast, and these respond poorly to movement. All right. So one of the things, one of the ways I remember it, I think about foveal vision, and I think parvocellular. Now, there's parvocellular other places, of course. All right, but this is what I think about mostly. This, I think more about black and white vision. So more peripheral. Okay, that helps me keep it straight. The coniocellular are going to provide information specifically about color. These are also small cells, or slow, small receptive size. These are similar in many ways to the parvocellular, except for they provide excuse me, information primarily just about color and blue yellow. Okay. In terms of the exam, you need to know that these exist. And you should have a rough idea about the apparent purpose of each. You don't necessarily need to memorize the entire table, though. Focus on what I highlighted. The magnocellular, these are going to detect large, low contrast objects and are important for movement. Okay, these don't respond to color. These are going to go to the magnocellular layers in the LGN. When you look in the retina, these are large cells. Now, because we're working with movement, these have to be fast, right? They process very quickly. These have a large receptive field size. That's good in some ways. They have good sensitivity, but low resolution. All right. And in terms of response to movement, obviously, the magnocellular cells are very good at it. All right. So we've talked a bit about the photoreceptor, bipolar cell, ganglion cells. With these ganglion cells, we've talked about M, P, K types. With the ganglion cells, we've talked about on center, off surround, and off center, on surround. Okay, so there's a lot going on even in the retina. Okay, so the ganglion cells kind of have a lot going on. I haven't written here amacrine or horizontal cells. Those I want you to know exist. I want you to know that they help with some processing. Horizontal, I do want you to know, are helpful for lateral inhibition. Okay, but that's really all you need to know about those cells. You need to know details about photoreceptor and transduction process. The bipolar cell, you need to know generalities, that it doesn't have action potentials, that changes in the membrane potential change how much neurotransmitter is released onto the retinal ganglion cells. The retinal ganglion cells you need to know some detail about. From each eye, we get the bundle of axons from the retinal ganglion cells going on to the brain. If the information doesn't get to the brain, you don't perceive it. Hanging off each eyeball is going to be an optic nerve. Okay, so this is from the eye to the brain, basically. Once we get to the brain, now some fibers are going to cross. We'll talk about that in a minute. But now those bundles of nerve fibers are called the optic tract. Okay, so optic nerve is from the eye up to the optic chiasm. That is where essentially it enters the brain. From there on, it's called the optic tract. The optic nerve carries information from one eye only. The optic tract carries info from both eyes. Okay, and when we draw out the connections, that'll be apparent. Now, all sensory information, except for olfaction, first goes to the thalamus. And that we talked about in one of the first chapters. The thalamus is complex and has many different subregions. One is called the lateral geniculate nucleus. I'm going to probably just end up calling it the LGN, lateral geniculate nucleus of the thalamus. Okay. This is a relay station. From here, information is going to come from the retinal ganglion cells. Those axons go all the way from the eye into the thalamus, where they synapse onto cells in the LGN. Those cells onto which they synapse then send axons to the primary visual cortex. And again, primary visual cortex is often called V1, often called striate cortex, and sometimes occipital cortex. Okay, these are all synonyms. I mentioned earlier that some basic processing starts here, but there's not enough computing power. Visual sensation is too complex for this primary visual cortex to do anything. So we're going to actually outsource, and we're going to outsource to different parts of the brain that are specialized for detecting motion, color, identifying objects. Okay, and we'll talk about a few of those regions as we go. Right now, I just want you to focus on what those different parts of the cortex do rather than memorizing just the names. All right. So this is a horizontal section through a cadaver. This stuff here is the brain. You can see here, this is gray matter of the cerebral cortex. This is white matter. You can see why we call it gray and white matter. It's distinct in a fresh brain. This is the skull. Here we have the eye. Okay, and in this particular section, this is an inferior view. So you are looking from the person's feet up. Okay, so that would make in this inferior view, we're going to have our eyes. You can see the lenses here. This right here is going to be the optic nerve from the eye up to this structure. Where the two optic nerves meet, this structure is called the optic chiasm. From here, you can see bundles of fibers leaving on their way to the thalamus, to the lateral geniculate nucleus. This is the optic tract. Okay, so optic nerve, optic chiasm, optic tract. If I cut the optic nerve right here, information from one eye will no longer get into the brain. If I cut the optic tract, though, I'm going to disrupt information flow from both eyes. Okay, and we'll see why that is in a moment. All right, someone's making a quick comment. It said that lateral inhibitions help us tell edges and boundaries, lines, right? So we can see objects. The horizontal cells are going to be part of, so we have multiple mechanisms to detect lines and edges. It's harder than you may think. So the lateral inhibition of the horizontal cells is part of that. So is the arrangement of the retinal ganglion cells. So is the receptive fields and the responses we have in the cortex. So we'll get ahead to that. Yep. All right. So this is a picture of the lateral geniculate nucleus. Okay, and if you look at the lateral geniculate nucleus, this is going to be a crescent violet stained section. You can see 1, 2, 3, 4, 5, 6 layers. These layers with the purple dots, remember crescent violet stains cell bodies. So this is where there's lots of cell bodies. In between, there's not air, there's not just space. It's actually axons, dendrites, things like that. So-called neuropil. Okay, so here we've got layers of connections and layers of cells. Okay, so again, we've got 1, 2, 3, 4, 5, 6. If you look closely here, Those are small cells. Okay, so the outer layers here are going to be the parvocellular layers. Down here, if you look what's labeled number 1 and 2, these cells are larger. These are going to be the magnocellular. Now, one thing about the arrangement, you can see we have an even number. One of these would be from the left eye. One would be from the right eye. Okay, so here the information from each eye is kept separate. Okay, so I honestly don't know from this picture. I don't remember whether this is a left or right lateral geniculate. I'm illustrating a concept here. All these cells are receiving information, say, from the left eye. All these cells would be receiving information from the right eye. This would be left eye, right eye, left eye, right eye. Okay, and we know that because we can inject tracers into the left eye and then look and see which of these layers lights up, which shows my marker that I injected into the eye. Okay, so we're keeping information from each eye separate here. The magnocellular, remember, magnocellular is going to be important for movement. It's got a large receptive field. Okay. Only about 10%, 15% of your retina ganglion cells are. So you're going to need less processing, right? The parvocellular, much more detailed, things like that, and a greater number. That's why we'll have more layers of those. All right, that's in the lateral geniculate. Again, this is just showing me, showing you another picture of the lateral geniculate nucleus. We can see our magnocellular layers. We can see our parvocellular layers. This is just a nicely stained section, so you can see those layers perfectly. All right. I mentioned the tracer. So in this, again, you can recognize the 6 different layers. Here they took a tracer and injected it into just one eye. And you can see that tracer only shows up in half the layers. And in the other layers, which presumably get info from the other eye, we don't see any tracer at all. Okay, so the point is your nervous system is keeping information from the left and the right eye separate. Okay. Again, I'm beating a dead horse here. You can look at this diagram in more detail if you would like to on your own. All right. So now things are going to get a little confusing. This is where you need to do some work. It really doesn't, it's not worth your time to try to like figure things out. Just memorize this. And the easiest way to memorize this is to just draw it out. The first time you draw it out, you're going to be confused. You're going to feel like you don't get it. That's normal. The reason I can whiz through it is because I've done this 1,000 times, probably more than that, right? The more you practice it, the better you're going to know it. Okay, so what we'll start with first is your visual field. Okay, so again, the visual field is everything you can see. We divide your visual field into a left and a right visual field. Okay. Now I'm going to ask you a question. Think about your left eye. Just consider your left eye here. Does your left eye see things in your left visual field? You can test that yourself. Close your right eye. Can you see on your left? Yeah. Is there information from your right visual field in your left eye? I can't see quite as much just because of the spacing of it, right? But I can see to my right with just one eye, with just my left eye. Okay, so both eyes are going to get information from both the left and the right visual field. Okay, so let's focus just on the left eye for right now. In this cartoon, everything in the yellow is going to be sort of this, I don't even know what color it is, bluish color here. Everything in the right visual field will be in yellow. Okay, and if you look again at the left and right visual field of the left eye and the right eye, just because of their positioning, there'll be some overlap, but there'll also be some that you can detect with your right eye that you can't detect with your left eye. Right, but the basic idea, we're going to have a left and a right visual field. Remember that light passing through the lens is going to be inverted and reversed. So if we look at anything in the left visual field, it's going to strike the right part of the retina. Okay, so here we've got the bluish color. Here on the retina, it shows which cells, which part of the retina that is being hit. Likewise, the right visual field is going to hit the left half of the retina. That's what we see in yellow here. Now, the eye gets information from the left and the right visual field. The cortex, though, the primary visual cortex, left primary visual cortex processes the right visual field. Okay, this is key. So if I destroy this right primary visual cortex, you won't be able to see anything on the left. I got to take a real quick break here. The dog is eating my homework, believe it or not. Hold on one sec.
And I would have to go.
No. Okay, dude. Sorry about that. Okay, so if we damage the right primary visual cortex, the entire entire left visual field will be gone. Likewise, if we destroy the left primary visual cortex, the entire right visual field will be gone. So literally, there will be a line down your vision. So the left part, let's say we destroy the left visual cortex. This is still intact, okay? So if we destroy this, your left visual field will be fine. But then there'll be sort of a line, and anything to the right of that line will be black. You won't be able to see anything. All right. So how do we go from your eye, which has information from both the left and right visual field, to only information from the contralateral, the opposite visual field in the primary cortex? Okay, so the optic nerve is going to carry information from both visual fields. Okay, and as that moves through the optic nerve, when we get to the optic chiasm now, our axons are going to follow different paths. Okay, so if we are talking about the right visual world, which is yellow, we're going to follow a path where we need to end up from the right visual world into the left primary visual cortex. Okay, so from the left eye, this information from the right visual field is going to stay in the same hemisphere of the brain. Okay, so that information is going to go through the optic nerve. When we get to the optic chiasm, it's going to stay on this side of the brain. Okay, so that information is going to get carried to the lateral geniculate nucleus, and from there to the primary visual cortex. The information from the left visual field, though, indicated in blue, has to get to the right primary visual cortex. So if we're in the left eye, we're on the left side of the body, that means we're going to have to cross sides. So what we do is our axons come through the optic nerve. When we get to the optic chiasm now, they cross to the contralateral side. And so these are going to cross over to the right lateral geniculate and then onto the right primary visual cortex. Okay. So to sort of help you see if you understand it, if I destroy the optic nerve, we cut the optic nerve, how is vision affected? Obviously, you won't be able to see anything out of your left eye. But if I cut the optic nerve, will you be able to see from the left visual field? My right eye still works. Does my right eye detect things from the left visual field? Yep. Does my right eye still detect things from the right visual field? Yep. So if I cut the optic nerve, I lose vision from one eye. I lose information. But I can still see both the left and the right visual fields. What would happen if I cut the optic tract? Remember, from the optic chiasm to the lateral geniculate nucleus, it's called the optic tract. What if I cut the left optic tract? Now information can't get to the geniculate or to this primary visual cortex. So what information do I lose? If we're on the left side, we're processing information from the right visual field. So if I cut this, I lose all information from the right visual field. Can I see out of the left eye? Yeah. Can I see out of the right eye? Yeah. But because this is cut, I can't see the visual field, the right visual field, from either eye. I got a couple questions. Let me check these out. Okay. Good. Here's just another diagram to show you the separation. This is something, again, I've already kind of walked you through it and explained it. I can do it a million times, but the only way for you to learn it is for you to actually draw it out. Okay, so after you watch this, take a blank piece of paper and draw this out a bunch of times. The type of question I'm likely to ask you, what would happen to activity in the right lateral geniculate nucleus if I cut the left optic nerve? So if we cut this nerve, we would lose about half the activity here, right? We'd lose this information, wouldn't make it in, so we'd lose half the activity. I might also ask you how vision would be affected. Okay, so you need to know these pathways. So when I ask you what happens when there's damage here, how your visual field would be affected. All right, so I'm going to jump back for a second to the retinal ganglion cells. Just again, to help you, the more you hear it, the better you remember it. Processing starts in the retina. Okay, there's very little processing in the lateral geniculate, but most of the processing is going to occur in the cortex. When we talk about the retinal ganglion cells, we're going to keep information from each of these ganglion cells separate until we get to the cortex. Okay, so parvocellular, we already talked about these having small cell bodies. These are near the fovea, details and colors. These go to the lateral geniculate nucleus. Magnocellular, large cell bodies. These are more in the periphery, large receptive field, really good for motion. Most of these project to the lateral geniculate nucleus. The coniocellular ganglion cells are small. They're all over the retina. These project to many areas, and we'll actually talk about so-called retinohypothalamic tract later when we get to circadian rhythms. All right. Now, when we go from these retinal ganglion cells to the lateral geniculate nucleus, we're going to follow pathways. And again, these pathways are not they're logically named. So those parvocellular cells are going to form the parvocellular pathway. Magnocellular will form the magnocellular, etc. Okay, so the pathways are just named by the type of retinal ganglion cell that they hook up to. When they get to the lateral geniculate, right, then you know that these are going to synapse onto those parvocellular layers. These will synapse onto those magnocellular layers. Now, when we get to the cortex, remember, mostly we've kept this information separate, and we keep it separate all the way to the cortex. Okay. Now, once that information gets to the cortex, we need to perceive it. We need to make sense of it. We're not going to go into details about how the cortex does that. One is we don't really know in detail. And second of all, that's beyond the scope of this course. Sensation and perception should cover that in more detail. All right. But from the cortex, we're going to talk about a mostly parvocellular pathway. So this carries information from those parvocellular retinal ganglion cells to other parts of the brain. This is going to carry information about details of shape. The mostly magnocellular is going to carry information to parts of the brain that are going to help us detect movement and then integrate vision with action. So let's say someone tosses a ball toward you. You want to catch it. It's always your best option if you can. So you're going to try to catch it. We need to integrate vision with movement. That requires a lot of brain power, a lot of integration, and so that'll occur in other parts of the brain. And then we also have a mixed pathway that has information from both, and that's going to deal with perceptions of brightness and color. Okay, so again, what these pathways are is important. If there's damage to the parvocellular pathway, a person will have difficulty processing details of shape. Okay, and so this is just another way of showing that. Don't worry about memorizing the brain regions. Okay, so if we look at the magnocellular pathway, remember these were the large periphery, good in low light, good at detecting movement, right? So that information is going to go from the primary visual cortex. It'll go to secondary visual cortex, and eventually to a part of the brain called MST or MT. This part of the brain, highlighted in green, is going to process movement and perception. Okay. Again, there's not enough processing power just in here. We need to specialize. So we're going to outsource that processing to this part of the brain. If there's damage to area MT or MST, what do you think the person has difficult doing? Here, they can see just fine. So they want to cross the street. They're not paying attention to the signals like they should. They see a bus. They can see the bus fine. They can recognize it as a bus. They can read the route number, everything. But they can't tell whether that bus is moving. They can't tell how fast it's moving, right? Not a good situation, obviously, if they want to cross the street. So damage to this part of the brain would lead to problems detecting movement. All right. We talk about a mixed, again, doing some color. This mostly parvocellular pathway is going to go to several different parts of the brain that are key for complex shape analysis. All right. When we are talking now about leaving the cortex, if we're looking at those pathways leaving, we can separate it into a dorsal stream and a ventral stream. Okay, remember dorsal. Where's dorsal? So if you have, here's how I always remember it. If I see a shark, right, I got my dorsal fin. So dorsal in this case would be the top of the head. Okay, so top of the head or back would be dorsal. Okay. Information leaves through a dorsal stream. Stream just means bundle of axons, okay? So this dorsal stream, information about where an object is or how it's moving is carried in the dorsal stream. There's also a ventral stream, okay, toward the bottom of the brain. This is information about what you're seeing. If there's damage to the ventral stream, or the parts of the brain that it goes to, we have something called visual agnosia. That means you can't ID an object by sight. So if I hold up a pen and ask you to identify it, you can do that. If a person has damage to their ventral stream, they can't. So I hold it up and I say, what is this? And they say, I don't know. If I hand a pen to them, though, and let them feel it, then they can identify it using tactile cues, though. Okay, so the problem isn't identifying objects per se. The problem isn't that they can't remember what a pen is. The problem is that they can't recognize the sight, the visual information, and pair it with what that object is because of damage to the ventral stream. There's a specific kind of agnosia. Agnosia in general means you can't identify an object. There's visual agnosia, which means you can't do it by vision. There's also tactile agnosia, which means you can't do it by touch. Okay, so agnosia is a general term. I'm talking now in this unit about the visual system, so we're talking about visual agnosia. All right, there's something called prosopagnosia. You may have heard of this. Some estimates say up to 10% of people may have a variation of this. To me, that's kind of unbelievably high, but it hasn't been well studied, so it could be that high. This is where a person has difficulty recognizing faces. So you can show a person a face and they are unable to identify even if it's somebody they know. And there's different degrees of prosopagnosia. Some people just have difficulty doing it. Some people are unable to do it, right? So it's kind of a continuum. I actually had a colleague who was unable to identify faces. She just couldn't recognize them as faces, and she couldn't identify people as individuals based on their face. So what she would actually do when you would come up to talk to her, she would use other cues to identify you. So we all have a different walk. So she could recognize people by their walk or by the shoes they wear or their height, other cues that she could recognize. But if she just looked at your face, she couldn't tell who you were from just your face. Okay. Now I'm going to spend some time talking about cortical cells. I'm going to talk about simple, complex, and hypercomplex. The nomenclature's changed a little bit. So when I was growing up, when I was your age, we called them hypercomplex cells. Now people call them end-stop cells. We're in a transition, so you should understand both. We're talking about the same thing. So a simple cell, remember, this is a cortical cell. So we are in V1, primary visual cortex. Okay, this is key, cortex. This represents the visual field of that cortical cell. And here, instead of just a single point of light, we're looking at a bar of light. Okay? A simple cell is going to respond to a bar of light in a specific orientation. Okay? Now, if that bar of light happens to be in the center, we get an increase in action potentials. Okay, so here the bar of light is in the centers. We get lots of action potentials. If that bar of light moves to the side, instead of excitation, we're going to get inhibition. If that bar of light is in a different orientation, so here we're at an angle, we're going to have some in the inhibitory areas, some in the excitatory area. The net effect of this, though, will be that this simple cell in the cortex won't respond to this. Likewise, if we illuminate the whole field, it doesn't respond. Okay, so if you think about this, this is going to be a way of helping us detect an edge. We can tell right where that bar of light is. When this cell increases firing, we know right in space where that bar of light is. When we're inhibiting firing, again, we still know where that bar of light is. We have some idea. In this particular simple cell, it won't respond to a bar of light at this angle. All right, but there are other simple cells that do respond. Okay, so this is one. There will be another one that looks like this. Okay, so another cell will respond. Okay, and actually, I think I told you, if you think about the retinal ganglion cells, right? If we have a center on, surround off, and we put a whole bunch of those together, you can see how that comes up with something that looks a lot like this, right? The centers of those cells correspond with this. The surrounds, right, correspond with these inhibitory regions. Okay, so again, by one simple cell getting information from many retinal ganglion cells, we can build more and more complex visual fields and responses. All right, so that's a simple cell. Again, this is just another diagram showing you the same thing. You can take a look at this on your own. I'm not going to go through this one. Now, in a complex cell, we're going to respond to a bar of light in a particular orientation and movement. Okay, so in this particular case, if there's a bar of light anywhere in the receptive field of the complex cell and it's in a specific orientation, we're going to get an increase in firing. We'll get action potentials. Okay, it doesn't matter where it is, right? As long as it's in that specific orientation, the cell is going to respond. And if this is moving, we get an even greater response. If the bar is at a different orientation, like here, we get a low response or no response. Okay, so here we're sensitive to a bar of light in a specific orientation. Okay, so again, just showing you what goes on here. Here we've got the whole receptive field. If we have a bar of light in that orientation, we get action potentials. If that bar of light is anywhere in the receptive field of that cell, we get an increase. Okay. All right. Now for your hypercomplex or end stop. Here we have a visual field or receptive field, sorry. Receptive field. Part of it is going to be on. Part of it will be off. Okay, so when there's no light, no stimulus present, this cortical cell will just spontaneously fire some action potentials. When we put a bar of light into it, it increases its firing rate. Okay, so if we have a bar of light in the on part, we increase the firing rate. Now, as this moves, as long as we stay in the on, we're going to get a strong response. Once we hit this off part now, right, we're going to have that strong response, but some inhibition. Okay, so what ends up happening is we get some firing of action potentials, but not as strong as it was as if we were in all on. Okay, and then as we move through and more gets into the off and less is in the on, the cell stops responding. And we get fewer action potentials, more inhibition. All right, so if you think about this, what this does, if we have a bar of light here, okay, and this cell starts firing, where's that bar of light? Well, it's somewhere in this area, right? We don't have a real precise location yet. As we move it, again, same response, right? Your brain can't tell the difference between these. All you know is there's a bar of light. Now, suddenly, when the response goes down, what just happened? We know that that bar of light crossed over into the off, right? So this is going to help us detect the edge of that bar of light. We're firing a lot. Okay, it's somewhere here. Once now suddenly we start to slow because we're getting into the off, we know right where the edge of that bar of light is. Right, and again, we can put these things together. It's going to help us detect edges, which is how we see objects. All right, so again, just another diagram. You can walk yourself through this. All right, for color perception, I'm going to be pretty quick and pretty brief here. What we can perceive, so when I say what we can see, what I can see, here I'm really talking about perception. Okay, so first we need to detect a stimulus. Once we detect it with receptors and all of that, the brain is going to put it together and we perceive it. Okay, so if we look at our vision, we're trichromats. So we have 3 pigments that give us color information. Really, we have 4 pigments, right? Because we also have black and white vision. All right, but we got 3 pigments. Those are going to allow us to differentiate different wavelengths of light. So we can differentiate green from this kind of orange, from this kind of gray. Other species that don't have it get some information, obviously, from the visual world, but not as detailed. All right. So a lot of times, do dogs have color vision? People get into arguments about this. Yeah, they have color vision, but it's not the same color vision that we have. Okay, so we have 3 cones. This is how we see this picture. Right, 3 cones allows us to fully enjoy the wonderful coloration of this guy's awful shirt here. Right, we can see and distinguish all different kinds of colors. Dogs have 2 cones. So if we look at 2 cones, what do they perceive? Well, do they have color vision, first of all? Yeah, we can see some colors. Do they have color the same way we do? No. Things are altered, right? Okay. So it's the cones that are going to allow us to differentiate wavelengths. Remember, each color is a different wavelength. So we can tell the difference between this wavelength and, say, yellow. A dog may not be able to do that. They can't distinguish different wavelengths of light, so they look the same. The more cones, the more photopigments you have, the better you're going to be able to see different colors. Humans are pretty good at seeing color. We're by no means the best. I'm trying to remember, I can't remember the precise number, but goldfish have something like 7 photopigments. Pigeons have more. So other species are much better able to differentiate wavelengths than we are. They see more colors than we do. Now, in terms of color blindness, we're not going to spend much time on it. I don't want you to spend time memorizing all the different kinds. But if we're lacking a photopigment, we are going to have some difficulty with color vision. So here is a photograph of someone with normal color vision. Again, you can see yellow, green, blue, presuming, of course, you have typical color vision. About 10% of men have some sort of color perception deficiency. Don't worry about these names here, but if we're lacking different cones, remember we have 3 short, medium, and long wavelength cones. Depending on which one we're lacking, we're going to view things differently. Now, do these people still have color vision? Yes. But is it typical? No, it's different, right? So if you're lacking these photopigments, again, you can see blue's okay, right? You can see yellow, that's okay. But things like green, you don't perceive very well. With this, you perceive different colors poorly, right? So this is supposed to be a blue shirt, but in this case, it would appear green. Instead of yellow, this appears kind of pinkish. All right, so these are what people would perceive if they lacked just one of the different cone photopigments. Pure lack of color vision, pure black and white vision is very, very rare. So when I say 10%, 10% of men have one of these. In terms of not being able to detect any color at all, that's exceptionally rare in humans. All right, so how do we perceive color? There are a few hypotheses. The bottom line is we don't have a wonderful handle on this. In my course, I'm going to stick mostly to the nuts and bolts, so I get off a little bit easy. Your sensation and perceptions instructor should go into more detail about this. One is the trichromatic or Young-Helmholtz hypothesis about color vision. And what that says is it's basically the ratio of activation between our cones that lets me perceive color. So let's say, I've been through this before, I'll go through it again. Let's just look at this, the long wavelength. what used to be called the red cones. All right, if I look at this, and let's say I have 80% activity, okay? That could be here or it could be here, right? 80%. So am I looking at kind of a green or am I looking at an orange? If I just have this, I don't know. I can't distinguish those two wavelengths, okay? Now, though, if we throw in another cone, let's say our reds are 80% and our green is near 100%. Okay, so red, I call it green. Medium is a preferred nomenclature now. All right, so if I'm looking at light and I have 80% red and 100% of green, well, 80% could be this or this, except my green has to be 100. There's only one place where my green is at 100 and my red is at 80. Over here, my green is what? 20. Okay, so if we have 80 and 100, it can only be one wavelength of light. So I'm looking at something here in the green. Okay, so that's the trichromatic or the Young-Helmholtz theory of color vision. Okay, that it's the ratio of activation of the three different cones that allows my brain to distinguish different wavelengths of light. And then when we have this wavelength of light, we perceive this color. There's something called the opponent process theory, which I think is a really cool theory. The Young-Helmholtz and opponent process theory are not incompatible with each other. Neither explains everything. Together, they may actually work together. They may both turn out to be correct. You're familiar with the idea of center on, center off. Okay, this is going to be completely different. Okay, so we're still talking about a retinal ganglion cell, but we're talking a little bit differently now. All right, so a retinal ganglion cell at rest is going to fire some action potentials, just spontaneous. Here, this represents the receptive field of a retinal ganglion cell. Okay, there's several different types. I'm just going to show you one here to illustrate the concept. In this particular retinal ganglion cell, if you shine red light, it's going to activate, it's going to turn on the center. Okay, so if we shine red light on the center, we're going to get an increase in firing. Okay, if we were to put green light in the outside, it's going to turn it off. So if we have just green light in the outside, that is going to decrease the firing rate. Okay. In this case, if we shine red on the whole visual field, we get an increase. Not as strong, but we get an increase. If we shine red light in the center, green on the surround, they kind of cancel out and we get no response. Okay, so here we talk about red and green. In terms of this, those are sort of opposite colors. So red is going to excite, green is going to inhibit. There's also blue-yellow. So instead of red and green, we'll have blue and yellow. And these will change. All right, so the way this hypothesis goes is anytime this ganglion cell increases its firing rate, anytime we see an increase in firing rate, you are going to detect red. Anytime there's a decrease in firing rate, you're going to perceive green. Okay. The number of action potentials isn't what's important. It's the change in action potentials. Okay, so increase equals perception of red. And although it's not shown here, a decrease equals perception of green. Okay, that's the basic concept showing you this one cell. There are other types of cells too, like I said, blue, yellow, that are going to help you understand. Now, some of the strongest evidence for this opponent process, I want you to spend about 10 or 15 seconds really staring at the red X. Okay, concentrate. Don't blink. Don't close your eyes. After about 30 seconds, I then want you to shift your vision over to the white square. Now, when you shift your vision over, you should see what's called an after vision. So that after vision, you're going to see an X. What color is that X? On my screen, at least, it is green. Okay, so how come? When I am staring at this, let's talk about that red green cell. Okay, when I'm staring at the red X, I increase firing rate. Okay, so I'm looking at the red X. The firing rate of that red green ganglion cell goes up. My brain perceives that as red, and I see the red X. Now, when I shift to the white here, what happens? Light's no longer striking that, right? No red light. What does that mean? A decrease in firing rate. What do you perceive when you see a decreased firing rate? The perception of green. Okay, so again, there's no stimulus here, right? There's nothing hitting your retina, nothing you should be seeing. Okay, that's why I emphasize what your body is sensitive to is changes in firing rate. Here, when you look at the red X, you're getting this going, you're getting it all excited, it's firing tons of action potentials. Okay, when I look over here, suddenly we stop firing action potentials. There's a decrease in your firing rate. And that, whenever we have that decrease here, your brain perceives that as green. Okay, so red and green are opposite colors. Do the same thing here. All right, so hopefully that yellow X or yellow plus turned into a blue plus. All right, same concept. Okay, so with the opponent process theory, red and green are opposite colors, blue and yellow are opposite colors. All right. So in terms of what I wanted to go over in vision, that's essentially the crux of it. I'm going to go ahead and stop recording now, and then I'll answer questions.